In this video, we're going to be talking about amino acids and pKa. By the end of the video, I want you to be able to draw any amino acid if I give you its R group. I want you to define the difference between pH and pKa. I want you to explain what pKa measures. I want you to explain the relationship between the pKa of a molecule, between the pH of the solution that molecule is dissolved in, and the protonation state of that molecule. How much of it is deprotonated, how much of it is protonated. And finally, I want you to describe why knowing the pKa of different parts of a protein is essential for understanding the optimal pH of that protein. So, amino acids are the monomers of proteins, and proteins are an incredibly diverse and pervasive macromolecule. In fact, whatever is not actually made of proteins within your cells probably required a protein to be made. Let me explain what that means. So first, lots of things within your cells are made of proteins. We've got defensive proteins like antibodies, we've got storage proteins like ovalbumin, transport proteins, hormonal proteins like insulin, contractile and motor proteins like actin and myosin within your muscles. So lots and lots and lots of different things within your cells are made of proteins. But there's also lots of non-protein things. There's nucleic acids, for example, DNA. But in order to make DNA, a cell needs to have these biochemical reactions that attach nucleotides together. Well, in order for a cell to do that, it needs to catalyze those reactions. And a biological catalyst, or I should say, most biological catalysts are themselves enzymes, are proteins. So what that means is even if you're looking at something that is not made of protein within a cell, you probably need an enzyme, a protein, to make that thing. For example, nucleic acids. So, like I said, the monomer of proteins are amino acids. There are 20 different amino acids, and they are identical in this group here. All of them have this alpha carbon, to which is attached an alpha amino group and an alpha carboxyl group and a hydrogen here. In that fourth position, that fourth covalent bond, is what is referred to as the R group of the amino acid, or the side chain of that amino acid. There's 20 different amino acids, and therefore there are 20 different side chains. We will look at them more later. There are several ways that you could name an amino acid. There is the full name, glycine. There's a 3-letter abbreviation, GLY, and there is a single letter abbreviation, G. As you can see, all amino acids have at least one carboxyl group and at least one amino group. And so it's absolutely essential that you refer to or review this idea that both carboxyl groups and amino groups are weak acids. When you put them into solution, they reach an equilibrium between the protonated state and the deprotonated state, between the protonated state and the deprotonated state. And make sure that you can apply each one of these terms to the two different states the amino group and the carboxyl groups take. Okay, I'll let you do that on your own. Now, one question I have, if we put the same concentration of a carboxyl group and an amino group into a solution and let it reach equilibrium, will we have the same concentrations of protonated and deprotonated group for the aminos and the carboxyls? Let's take a look. All right, so let's make a hypothetical situation here. Let's take a molecule that has a carboxyl group on it, and let's dissolve that molecule into an aqueous solution. So we'll take 1 molar of this molecule, and it has a carboxyl group, and we put it into an aqueous solution. Now we know that a carboxyl group is a weak acid and not a strong acid. So what that means is that when equilibrium is reached, some of that molecule that we've dissolved into the solution will become deprotonated, but not all of it. So we'll end up with something like this. Some of it will deprotonate, and some of it will remain protonated. So that's our protonated, and this is our deprotonated. Okay. Now let's take another aqueous solution, and to this one, let's add 1 molar of a molecule that has an amino group. And that will then reach equilibrium like this. So an amino group is also a weak acid, meaning when you put it into a solution, it will become deprotonated, not all of it. It's not a strong acid, okay? It's a weak acid. It'll donate its proton. And so here we have in equilibrium some of the protonated state, some of the deprotonated state, okay? Here is the important question that I want you to think about. If we put 1 molar of the protonated carboxyl group into a solution, some of it is going to break down, right? It's going to deprotonate into the proton here and the conjugate base, the deprotonated form here. Same thing here. You add 1 molar, some of that 1 molar is going to deprotonate. So the important question that we're going to address here is, does a carboxyl group have the same affinity for that hydrogen as the amino group has for its hydrogen? Meaning, which one of these two drops off and becomes deprotonated easier, the carboxyl group or the amino group? Basically, can different molecules have different affinities for their protons? And the answer is absolutely. Turns out, and we'll see, we'll assign numbers to this, when you put a carboxyl group into a solution, more of it will deprotonate than if you put an amino group into solution. So that by the end of this, a carboxyl group might almost be something like 0.8 molar deprotonated, where an amino group may be something by the end, when we reach equilibrium, something like 0.4 molar. Now those are just totally made up numbers, but that's the point I'm trying to get across here, is that just because you have 2 molecules that are deprotonatable doesn't mean that they deprotonate at the same rate, okay? That some molecules have a higher affinity to their protons, to their hydrogen ions, and some have a lower affinity, okay? Now that affinity, that characteristic of a molecule, that characteristic that defines how well it holds on to its proton or how easy it drops that proton when put into an aqueous solution, the value that we are going to look at and that we're going to work to define over the next couple slides is called pKa. And that is a measure of a molecule's affinity to its, its proton, okay? So that's what we're going to work to define over the next few slides. The dissociation or ionization constant, or Ka, reveals a molecule's affinity for its proton. Let's see what I mean about that. Here is a protonatable group. Here's a weak acid. All right, we put it into solutions. Some of it stays protonated, some of it deprotonates. We let it reach equilibrium, and we look at the concentrations of each one of these groups here, and we slap it into this equation here. And what we get is something called the Ka, the ionization or dissociation constant. And notice, because the concentration of the protonated group here is in the bottom, right, this is what's going to give us an idea of how strong an affinity a molecule has for its proton. And that's what I'm asking here. Which group will have the stronger affinity to its proton? Well, it's going to be the group that when we reach equilibrium will have most of its molecule being protonated here. It's still holding on to its proton, which means, since it's the denominator here, it's going to be the group with the lowest Ka. So glycine's amino group has a Ka of 2.5 times 10 to the minus 10th molar. Glycine's carboxyl group, alpha carboxyl group, has a 4.5 times 10 to the minus 3 molar Ka. 10 to the minus 10th is a much smaller number than 10 to the minus 3rd. That means that it's the amino group that has a higher concentration of protonated at equilibrium, and so that means that it's the group with the lowest Ka. Now, turns out saying 2.5 times 10 to the minus 10th molar is the Ka of the amino group, that's a mouthful. And so what scientists have done is they've just sort of transformed that number. They said, okay, let's just talk, instead about Kas, let's talk about pKas. And the pKa is simply the negative log of the Ka. That makes it much easier to talk about. So instead of saying something like the Ka of glycine's amino group is 2.5 times 10 to the minus 10th molar, we can simply say, oh, the pKa of glycine's amino group is 9.6. Much easier to talk about. The Ka of glycine's carboxyl group is 4.5 times 10 to the minus 3rd. Well, that's a bit windy. Instead, we say, pKa of glycine's carboxyl group is 2.34. But because we've got this sort of inversion where the pKa is the negative log of the Ka, what that means is even though the lower Ka means the higher affinity to the proton, now the pKa, what we want is the higher the pKa, the higher affinity to its proton. Okay, so the group that has the higher pKa has the higher affinity to its proton. Ka is a constant, which means when we reach equilibrium here, if we start messing around with the concentration of the protonated group or the deprotonated group or the protons, this reaction is going to change. This equilibrium is going to move left or right. So what would happen if we take this equilibrium and we increase the pH? What would happen if we take this at equilibrium and we decrease the pH? Let's take a look at that. Now, the fact that the dissociation constant is a constant, and because of Le Chatelet's principle, or the equilibrium law, there's a really important consequence. So if we have a acid, a weak acid, and it's at equilibrium with its conjugate base, all right, then if we do anything to affect that, that equilibrium, it'll react. And what I want to look at is what happens when, so we've got this and it's dissolved in a solution, and that solution is at a particular pH. It has a particular hydrogen ion concentration. And so now let's see what happens when we change the pH. What's going to happen to this equilibrium? So let's say we are looking at the pH and we increase it. So we are increasing the pH. So what we need to know, remember, pH is simply a way of expressing the hydrogen ion concentration. So if we're increasing the pH, what are we doing to the hydrogen ion concentration? Higher pH, lower hydrogen ion concentration. So we are lowering the hydrogen ion concentration, which means we are removing hydrogen ions. So how is this equilibrium going to react if we are removing product? Well, it moves in this direction. More of this deprotonated species here is going to break down to supply more protons and therefore more deprotonated versions of itself. Now it'll go in the other direction too. Let's take a look. Let me bring this back to our original state. And now let's see if we decrease the pH. Let's see what happens. We got the pH and now we're decreasing it. All right, so a decrease in pH. What does that mean for the hydrogen ion concentration? The lower the pH, the higher the hydrogen ion concentration. And so what that means is we're adding protons. We're increasing the concentration of protons in solution. That is going to affect our equilibrium. Some of those protons are going to bind some of this conjugate base here, this deprotonated state, and we are now going to drive that reaction in the right to left, in the reverse direction here. So we are going to be using up some of the conjugate base, some of the deprotonated. It's going to become protonated, and this is going to increase in concentration in solution. So what this means is that the pH of a solution will change how much of a molecule is in the protonated state and how much of that molecule is in the deprotonated state. Okay, so what we immediately see then is that there is a relationship between the pKa of a molecule that defines its affinity, its inherent affinity to its proton, the pH, right, the proton concentration of the solution, and the concentration of deprotonated versus protonated form of that molecule. And all of that is defined by this handy-dandy equation, the Henderson-Hasselbalch Equation. pH of the solution in which your molecule is dissolved, the pKa of that molecule, both play into the concentration, the relative concentrations of the deprotonated and the protonated form. And the derivation of this is very straightforward. It's straight from the dissociation constant. Here's the dissociation constant. We know we can express Ka as pKa. We know we can express the hydrogen ion concentration as pH, and so we can take this and turn it into this. And then we can take this, do a little bit of math magic, and turn it into the Henderson-Hasselbalch equation. You won't be required to do this derivation, but I just want you to know that it's not very far removed from what you already know. Now, when we look at the Henderson-Hasselbalch equation, there's something interesting that happens when we have a situation in which the pH of a solution that a molecule is dissolved in is equal to the pKa of that molecule. Let's take a look at that. So there's something special when the pH of a solution that a molecule is dissolved in is equal to the pKa of that molecule. Okay, so here we have a molecule. It has a carboxyl group on it. Let's say the carboxyl group has a pKa of 3, and we dissolve that molecule into a solution, and the pH of that solution we've adjusted, right? We've done it ourselves by adding some acids and some bases. We've adjusted that pH to 3. So what happens in this situation? And it's really important to understand what happens. In order to figure this out mathematically, we have to go to the Henderson-Hasselbalch equation, the equation that defines the relationship between pKa, pH, and the proportion of deprotonated versus the protonated state of a molecule. And the Henderson-Hasselbalch equation looks like this. So the pH of the solution in which the molecule is dissolved is equal to the pKa of the functional group that we're talking about, plus the log of the concentration of deprotonated state over the concentration of the protonated state. Okay, so what we're looking at is what happens. What is the special thing that happens when the pH of the solution is equal to the pKa of the protonatable group that's dissolved in that solution? Well, let's do some math magic here. This is 3, and this is 3. Then when we subtract this from this, we get 0. 0 now is equal to the log, the log of the concentration of deprotonated state divided by concentration of the protonated state. Now, the log of what equals 0? Okay, or you can basically say, what's the anti-log of 0? So the log of 1 equals 0. So what that means is when the pH of the solution is equal to the pKa of the protonatable molecule that's dissolved in that solution, we get this special relationship. The concentration of the deprotonated state divided by the concentration of the protonated state is equal to 1. And even more importantly, let's write it that way. What that means is the concentration of the deprotonated state of that molecule is equal to the concentration of the protonated state. This is a important kind of benchmark to think about when we're thinking about protonatable molecules and the effect that pH of the solution that they're dissolved in, the effect of pH on them. Okay, so let me reiterate. When you dissolve a molecule that has a pKa into a solution that the pH is equal to that pKa, then what you know is at that point, when the pH of the solution is equal to the pKa, the concentration of deprotonated forms of that molecule is equal to the concentration of protonated forms of that molecule. Okay, so from what we've learned then, we can kind of use that as a benchmark for getting a feel for what pKa means. So here we have our solution that we're adjusting the pH of this solution. Let's say right now the pH is 4.5. And we have this weak acid, HA, in that solution. And it happens that this weak acid has a pKa of 4.5. So we're at a situation here where the pH of the solution, 4.5, is equal to the pKa of the molecule, 4.5. So at that point, we know that half of the molecules are protonated and half of the molecules are deprotonated. All right, so now, if we then adjust the pH so that we are increasing the pH to 5, to 6, to 7, by reducing the concentration of hydrogen ions, what we're doing is we are getting rid of hydrogens. That means this equilibrium shifts in the forward direction, which means we start having less and less protonated and more deprotonated. More deprotonated, less protonated. And we're going to get to a point where eventually, when we are at a pH that is 2 units greater than the pKa, so at 6.5, 99% of those molecules are going to be deprotonated. Let's go in the reverse direction. So, all right, we're back at pH is equal to the pKa. We got 50%, 50% protonated and deprotonated, and now we're adding acid, like a strong acid. We're reducing the pH. We're increasing the hydrogen concentration. That means if we're increasing the hydrogen concentration, we're driving this equilibrium to the left in the reverse direction. That means we have less and less deprotonated, more and more protonated. Okay, until we get to a point, right? We're going to eventually get to a point where we are 2 pH units less than the pKa. So we're down to 2.5. At that point, 99% of those molecules are going to be protonated. Well, where did I get this number? This 2 units below or 2 units above the pKa, resulting in 99% protonated or 99% deprotonated. Well, I just did it via the Henderson-Hasselbalch equation. So let's take a look. We're looking at a situation in which the pH is at pKa minus 2 units. So let's define that. The pH is at a situation where it's pKa minus 2 units. We've got our pKa, that doesn't change, and then plus the log of the deprotonated over the concentration of the protonated state. All right, let's remove pKa, subtracting here, that means we have minus 2 equals the log of the deprotonated concentration over the concentration of the protonated. And then if we take the anti-log of both sides, we get 1 over 100 equals the concentration of the deprotonated divided by the concentration of the protonated. So that means for every 1 unit of deprotonated, there's 100 units of the protonated. If we then want to get to the percentage of protonated, we simply take the amount of protonated, 100, divided by the total, 100 plus 1. So 101 times 100, 99% of the molecules will be protonated when we have a situation where the pH is equal to the pKa minus 2 units. So that's the math that led to these 2 benchmark situations here that you should absolutely remember. All right, finally, let's take a look at why this is relevant for biochemistry, why this is relevant for what you are going to be learning. Knowing the pKa of the functional groups of a protein is essential for understanding the structure of the protein, understanding the optimal pH of that protein. Let's take a look at an example. I want to demonstrate why proteins have an optimal pH and why proteins will denature if you stray too far one way or the other from the optimal pH. So what we have here is a hypothetical protein. So this is a chain of amino acids, and 2 of the amino acids in this protein are lysine, and the amino group of lysine is shown here. That'll be the amino group on the R group. And aspartate. So this is the carboxyl group on the R group of aspartate. Now this amino group here in lysine has a pKa of 10.53, and this carboxyl group here has a pKa of 3.65. And let's assume that this is a protein within our cells that has an optimal pH of about physiological pH. So pH 7.3 or something like that. Okay. Now, when the pH is 7.3, let's take a look at how many of those lysines are going to be protonated. Let's see how many of those carboxyl groups, those aspartates, are going to be deprotonated. So let's focus on the lysine. The current pH of the cell is 7.3. And the pKa is 10.53. So that means the pH is at least 2 units less than the pKa. Okay, 2 units less than the pKa. And based on what we learn, when that's the case, when the pH is at least 2 units below the pKa, we know that 99.99% of molecules are going to be protonated, and because it's an amino group, it has a positive charge. Good. So we have it written up correctly here. Let's take a look at the carboxyl group. Now the pKa of that carboxyl group is 3.65. The pH is 7.3. So we have a situation here where the pH is at least 2 units above the pKa, and that means, again, you'll have to go and review the lecture, that means that 99.999% of the molecules will be deprotonated. In the case of a carboxyl group, that means it's going to carry a negative charge. Good. So what that means is at pH 7.3, most of the 99.9% of the amino groups are going to be positively charged, 99.9% of the carboxyl groups are going to be negatively charged, and we get this beautiful ionic interaction that forces the protein to fold in this way here. And it's this fold in the protein that is necessary for the proper structure and therefore proper function of this protein. Beautiful. Everything's working as it should as long as we are at the optimal pH. Now what we're going to do is we're going to take a look at what happens when we increase the pH and decrease the pH. So let's take a look at when we decrease the pH. So let's see here. If we take the pH and we decrease the pH by adding acid to the solution, what are we doing to the hydrogen ion concentration? We're increasing it, right? So we've got these protons, we're just increasing, okay? As we increase the concentration of protons, what's going to happen? Well, our NH3 plus is already protonated, so adding more protons, so decreasing the pH from 7.3 to 6.3 to 5.3, it's not going to affect the amino group. It's already protonated, cannot get any more protonated. But take a look at this. As we reduce that pH, maybe to 6.3, and then maybe to 5.3, maybe to 3.3, and so on and so forth. We're adding more and more protons, and what's going to start happening is this carboxyl group is going to become protonated and lose its charge. In this state here, this carboxyl group, now that it's protonated, cannot interact ionically with this amino group here. We're going to lose this fold. And we even have a good idea of when this is really going to happen. It's going to happen at a situation really where the pH that we are bringing this down to is 2 units less than the pKa of aspartate. So once we get down to 1.65, nearly 99.9% of these carboxyl groups are going to be protonated because we have such a low pH, such a high concentration of hydrogen ions that even carboxyl groups that don't usually hold on to their protons very well, even they are going to be protonated. So at this super low pH, let's say pH 1.65, we know our amino group is going to be protonated. It was protonated a long time ago. But we now know, since we're 2 units here, that almost 100% of our carboxyl groups are going to be protonated as well. We no longer have that ionic interaction, and that protein is going to denature. Now, let's look at the other side. Let's look at what happens when we take that pH and we start to increase it. We start to look at that proton concentration, and it's decreasing. So now what we're doing is we're removing protons, we're removing protons, we're removing protons. We are, right, increasing the pH, and eventually, all right, we're going to get back to our optimal pH. Beautiful. 100%, almost 100% of the carboxyl groups have become deprotonated again. They've got a negative charge. Those amino groups are all positively charged because they're all protonated. Beautiful. But now let's keep going. Let's keep increasing that pH. So now that we go to 8.3 and then we go to 9.3, well, as we do that, we're taking away protons, taking away protons, taking away protons. Well, that's not going to affect the carboxyl group at all. We've already taken away all of its protons right around pH 7. That was not a problem. But as we keep increasing, eventually what's going to happen is we're going to start to lose our protons from this amino group of lysine. And when we take away that last proton, we no longer have a charge. And now that we don't have a charge here on this amino group, we can no longer have this ionic interaction, and we're going to unfold that protein. It's going to lose its structure, and it's going to lose its function. On top of that, because we know the pKa of this amino group here, we know when this is going to get really bad. It's a point at which the pH is 2 units above the pKa. So when we bring that pH all the way down to, to 12.53, we will know almost 100% of these amino groups will also be deprotonated, and therefore we will not be able to have an ionic interaction with this negative charge on the carboxyl group, and the protein will unfold. It will denature. It will no longer function. So hopefully what you've got from this is that proteins have a optimal pH, and that's going to be the pH where all of the various weak interactions can occur. However, if you stray from that pH, if you go too high, if you go too low, you're going to start to mess up the ionization states of some of these important R groups. You're going to stop these weak interactions from occurring. The protein is not going to be able to fold properly, and it's going to lose its structure and therefore its function. All right, so hopefully now you have a good feel for these learning outcomes, that you can draw an amino acid if I gave you an R group. You can define the difference between pH and pKa and define what pKa measures. That you can define and explain the relationship between the pKa of a molecule, the pH in which that molecule is dissolved, and what effect all of that has on the protonation state of that molecule. How much of it is protonated, how much of that is deprotonated state. I hope now that you can describe why knowing the pKa of the different parts of a protein is important for understanding the optimal pH of that protein, or why that protein has an optimal pH.